Streptococcus bacteria exhibit various hemolytic patterns, including alpha, beta, and gamma hemolysis. Within the alpha hemolytic category, we find virulent streptococci and Streptococcus pneumoniae. Alpha hemolysis means partial oxidation of red hemoglobin to green methemoglobin. In contrast, Streptococcus gallolyticus and Enterococcus fall under the gamma hemolytic category, where there is no oxidation. And the beta hemolysis encompasses 20 groups based on the Lancefield classification system, where there is partial oxidation resulting in yellow color. The Lancefield classification system categorizes beta hemolytic streptococci into different groups based on the presence of specific cell wall antigens known as Lancefield antigens. These antigens are designated by letters A to V, excluding I and J. The two main groups within the Lancefield classification are Group A and Group B streptococci, which are significant pathogens in human infections. Group A streptococci, that is Streptococcus pyogenes, are responsible for a wide range of infections in humans, including pharyngitis, skin and soft tissue infections, scarlet fever, rheumatic fever, and invasive diseases such as necrotizing fasciitis and streptococcal toxic shock syndrome. Group B streptococci, that is Streptococcus agalactiae, are a leading cause of neonatal sepsis, pneumonia, and meningitis, particularly in newborns and infants. Group B streptococci can also cause infections in pregnant women, elderly, and immunocompromised individuals. Other Lancefield groups, such as Group C, D, and G, include various species of beta hemolytic streptococci that may cause infections in humans but are generally less common pathogens compared to Group A and Group B. Group A streptococci are classified using two typing methods: Griffith typing and EMM typing. Griffith typing relies on the M protein, while EMM typing is based on M gene. To differentiate between Streptococcus pyogenes and Streptococcus agalactiae, a bacitracin test is conducted. In this test, colonies of Streptococcus pyogenes are sensitive to bacitracin and die off, while those of Streptococcus agalactiae remains unaffected, showing resistance. Another test is the PYR test. The PYR test, short for pyrrolidonyl aryl amidase test, is a biochemical assay utilized for identifying specific bacterial species, primarily within the Streptococcus and Enterococcus families. This test relies on the enzymatic activity of these bacteria to break down a substrate known as pyrrolidonyl beta naphthalamide into a compound that elicits a color change. Streptococcus pyogenes and Enterococcus possesses an enzyme, that is L-pyrrolidonyl aryl amidase enzyme, enabling it to hydrolyze pyrrolidonyl beta naphthalamide and generate beta naphthalamide as a byproduct. Upon the addition of methylaminocinnamaldehyde, the solution turns red, indicating a positive result. Breaking down the term Streptococcus pyogenes provides insight into its characteristics. Strepto signifies chains. Coccus denotes their round shape. Pyo indicates pus, and genes suggests forming. Thus, these bacteria are typically round shaped and arranged in chains, known for their ability to induce pus formation. Streptococcus pyogenes possesses numerous virulence factors, including its capsule, which is unique for being composed of hyaluronic acid, while others typically have a polysaccharide capsule. Within its cell wall, specific antigens and outer membrane proteins such as M, T, and R are present. The M protein, in particular, serves as the basis of Griffith typing. Additionally, it produces various enzymes and toxins such as streptolysin, streptokinase, hyaluronidase, DNase, and streptococcal pyrogenic exotoxin. These factors collectively contribute to the pathogenicity of Streptococcus pyogenes. Streptolysin refers to a group of toxins produced by Streptococcus bacteria that can cause the lysis or destruction of red blood cells. There are two main types of streptolysin: Streptolysin O and Streptolysin S. Streptolysin O. This is an oxygen labile, oxygen sensitive enzyme that is active in the absence of oxygen. It can cause complete hemolysis of red blood cells, resulting in deeper hemolysis, and is involved in the pathogenesis of diseases caused by Streptococcus pyogenes, such as streptococcal, pharyngitis, and skin infections. It is stable in serum and antigenic, exhibiting properties that are lethal, cardiotoxic, and leukocidal. Whereas streptolysin S is stable in the presence of oxygen and is active even in the presence of oxygen. It is responsible for the partial hemolysis of red blood cells, resulting in surface hemolysis, and contributes to the characteristic beta hemolytic appearance of Streptococcus pyogenes on blood agar plates. Streptolysin S is soluble in serum and it is non-antigenic and it is only leukocidal. These toxins play a role in the pathogenicity of Streptococcus bacteria by aiding in tissue destruction and invasion of the host immune response. Streptolysin, known for its fibrinolytic properties, is an enzyme produced by certain strains of Streptococcus bacteria. Interestingly, Streptococcus equisimilis, that is group C Streptococcus, is utilized in the therapeutic applications. It's streptolysin. It has therapeutic application. Next is hyaluronidase, and it is another notable enzyme that aids in the breakdown of connective tissue. Its role involves the degradation of hyaluronic acid, a crucial component that maintains the tight linkage between our cells of connective tissue and blood vessels. Next is DNase. It is produced by the Streptococcus pyogenes, serves as a protective role against neutrophil extracellular traps, that is nets. Nets are structures formed by neutrophils to trap and kill bacteria. But this DNase helps the bacteria evade these traps by breaking down the DNA within them, allowing the bacteria to escape and proliferate. Next is streptococcal pyrogenic exotoxins. They are a group of toxins produced by certain strains of Streptococcus pyogenes. And they are classified into different types, including types A, B, and C, based on their antigenic properties. These toxins are secreted by Streptococcus pyogenes during infection and can have a range of effects on the host immune system and tissues. The primary effects of SPEs include superantigen activity. SPEs can stimulate a large number of T cells by cross-linking MHC class II molecules. This leads to the activation of a large number of T cells, causing a massive release of pro-inflammatory cytokines. This excessive immune response can result in systemic effects, including fever, shock, and organ damage. Next is erythrogenic toxin activity. Certain SPEs, such as streptococcal pyrogenic exotoxin A, also known as erythrogenic toxin, are responsible for the characteristic rash seen in scarlet fever. This toxin causes the rash by inducing a hypersensitivity reaction in susceptible individuals. Another effect is the tissue destruction. SPEs can also contribute to tissue damage and necrosis at the site of infection. They can disrupt cell membranes, leading to cell death and tissue destruction, which can exacerbate the severity of infections caused by Streptococcus pyogenes. Overall, streptococcal pyrogenic exotoxins are important virulence factors that contribute to the pathogenesis of Streptococcus pyogenes infections by modulating the host immune response and causing tissue damage. Their activity underscores the importance of targeting these toxins in the development of the therapeutics and vaccines against streptococcal infections. Apart from the well known conditions like scarlet fever, toxic shock syndrome, and necrotizing fasciitis, Streptococcus pyogenes can also trigger other health issues. Scarlet fever is identifiable by its distinctive blanching, papular rash, often described as resembling sandpaper. Pharyngitis, commonly known as a sore throat, is characterized by painful and tender cervical lymphadenopathy and typically lacks coughing as a symptom. Necrotizing fasciitis, often termed flesh eating bacteria, is an urgent soft tissue infection caused by Streptococcus pyogenes. This condition involves rapid degradation of fascia, subcutaneous tissue, and muscle due to toxins released by the bacteria. Symptoms include high fever, intense pain, swelling, redness, and rapidly expanding tissue necrosis, sometimes leading to purple colored skin. Early detection and aggressive treatment are crucial in managing this life threatening condition. Streptococcus pyogenes can also cause various skin infections such as erysipelas, cellulitis, and impetigo. Well, erysipelas and cellulitis both appear as painful red lesions. They differ in appearance and depth of skin involvement. Erysipelas presents as raised and well demarcated lesions, primarily affecting the superficial skin, that is epidermis, whereas cellulitis is characterized by non raised lesions that are not well demarcated and involve the deeper dermis. It's important to note that impetigo typically presents with an itchy, honeycomb colored scab on the skin. Streptococcus pyogenes can also trigger post infectious sequelae, such as acute rheumatic fever, which is caused by the presence of M protein in the bacteria. This protein bears resemblance to glycoproteins found in human cells. When the bacteria invade our body, our immune system mistakenly identifies our cells as foreign and mounts an immune response, leading to acute rheumatic fever. This phenomenon is known as molecular mimicry and falls under the category of type 2 hypersensitivity reactions. Symptoms of acute rheumatic fever can be remembered using the mnemonic Jones, which stands for joint inflammation, heart damage, nodules in elbow, knees, and joints, erythema marginatum, and Sydenham's chorea, characterized by rapid hand movements. Another condition, post-streptococcal glomerulonephritis, may occur following impetigo. It is a type 3 hypersensitivity reaction in which residual antigens from Streptococcus pyogenes in the bloodstream bind to antibodies, forming antigen antibody complexes. These complexes are then deposited in the basement membrane of glomeruli, leading to activation of the complement system and recruitment of neutrophils in the glomeruli, resulting in kidney damage. For diagnosis, a throat swab and blood agar tests are conducted. Additional biochemical tests include the catalase and oxidase tests, which yield negative results. As previously mentioned, the positive results of the bacitracin and PYR tests aid in the identification. Selective media such as PNF containing antibiotics and liquid media exhibiting granular turbidity are also employed. And as transport media, Pike's media is used. Treatment typically involves penicillin administration, as Streptococcus pyogenes has not shown resistance to it. However, for individuals allergic to penicillin, erythromycin can be prescribed as an alternative. Let's summarize. Streptococcus pyogenes. It is a group A beta hemolytic bacteria. It has various virulence factors such as capsule, which is made up of hyaluronic acid. And in its cell wall, it has antigen and outer membrane proteins such as M, T, and R. It also has enzymes and toxins such as streptolysin, streptokinase, hyaluronidase, DNase, and streptococcal pyrogenic exotoxins. Streptolysin, they are of two types, streptolysin O and streptolysin S. Streptolysin O is oxygen labile and it can cause complete hemolysis, resulting in deeper hemolysis. It is responsible for streptococcal pharyngitis and skin infections. It is stable in serum, it is antigenic, and it is lethal cardiotoxin and leukocidal. Whereas streptolysin S, it is oxygen stable, can cause partial hemolysis resulting in surface hemolysis, and it is soluble in serum, it is non-antigenic, and it is only leukocidal. Whereas SPEs, they are of three types, A, B, and C. A and C are bacteriophage mediated. B is chromosomal mediated. And this A and C, they have super antigen activity, which stimulate a large number of T cells by cross-linking MHC class 2 molecules, causing toxic shock syndrome. It also has erythrogenic toxin activity, which is responsible for scarlet fever. And it is also responsible for tissue destruction, causing necrotizing fasciitis. As for the signs and symptoms, for scarlet fever, there is sore throat, sandpaper rash, strawberry tongue, and fever. For pharyngitis, there is fever, painful swallowing, no cough, and tonsils. For necrotizing fasciitis, there is fever, it is painful, and there is purple skin. Next is erysipelas. The signs and symptoms for erysipelas and cellulitis is very important to differentiate between both of them. Erysipelas is raised, well demarcated, painful red lesions which affects the superficial epidermis. Whereas for cellulitis, it is not raised, not well demarcated, but it is painful red lesions and it affects the deeper dermis. As for impetigo, it is itchy, honey colored scab on skin. Next is acute rheumatic fever. For this, the mnemonic is Jones and the symptoms are joint inflammation, heart damage, nodules, erythema marginatum, and Sydenham's chorea. As for PSGN, that is post streptococcal glomerulonephritis, the symptoms are facial edema and dark red urine. Now, for diagnosis, throat swab, blood agar, selective media like PNF, and liquid media are used. And the catalase test and oxidase test, they both are negative, whereas bacitracin test and PYR test, both are positive. For treatment, penicillin is prescribed. But in case of allergy to penicillin, erythromycin is prescribed. And the transport media is Pike's media. If you found this information valuable and insightful, we would greatly appreciate it if you could show your support by giving this video a thumbs up and subscribing to our channel. Additionally, for quick reference and summarized notes, be sure to visit our Instagram page. You can find the link in the description box below. Thank you for tuning in and watching. Your support means a lot to us.